Hello, everyone, and welcome to implementation of an automated diagnostic WES and WGS workflow for multi-sample types using Covaris on the Hamilton Microlab Star Liquid Handling Platform. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more about our sponsor, please visit covaris.com. We do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation, and you can submit any technical issues there as well, and we'll help you with those via one-on-one. I'd like to now welcome our speakers, Tom Hofste, the head of Core Facility Genome Diagnostics Laboratory, Human Genetics Department, Radboud University Medical Center, and Madan Ambavaram, I apologize, Ph.D. Senior Application Scientist at Covaris. For a complete biography on our speakers, click the biography window on the left side of your screen. Now, Madan, you can go ahead and begin your presentation.
Hello, everyone. Thank you for the warm introduction. My name is Madan Ambavaram. I'm the Senior Application Scientist at Covaris. It gives me immense pleasure to present today alongside Tom Hofste, Head of Core Facility at Radboud University Medical Center, Madan. I would like to take this opportunity to discuss next-generation sequencing sample prep solutions using Covaris proprietary technology, adaptive focused acoustics, AFA in short. Covaris solutions for the NGS space. Let me begin by sharing the Covaris vision. Our global solution mission is straightforward and impactful. We aim to help people worldwide by delivering sample prep solutions that enable better information from biomolecules to improve and save lives. Covaris AFA technology provides comprehensive integrated workflows across genomics, proteomics, and beyond. Our technology enables highly efficient extraction and processing of biomolecules from a wide variety of sample types, such as you can see here in the left side, cells, fresh or frozen tissues, FFPE, blood, and saliva. This approach ensures robust, reproducible results that streamlines workflows across diverse omics applications, which includes genomics, transcriptomics, and protein analysis. This framework is our commitment to advance science across all fronts, catalyzing discoveries and supporting our customers to develop life-changing therapeutics and diagnostics. Speaking about Covaris workflow focuses, we constantly improving performance, economics, and ease of use. And these three pillars are the foundations of our focus and dedication to the genomics community. The claims presented, and you are seeing here, to name a few, for example, optimized analyte yield across samples, simplified QC processes, and workflow ease of use for automation. These claims are backed by data and real case studies with customers and collaborators, some of which I will be sharing in the upcoming slides. Covaris sample prep solutions and problem-solving strategy. As we transition to a more solution-oriented approach, let's take a comprehensive look at our capabilities. On the left, you will see Covaris offerings specifically tailored to meet diverse customer needs across various sample throughput ranges. Our portfolio spans instruments, consumables, and automation-compatible platforms, each designed to serve a wide spectrum of laboratory requirements. In the center, under the right section, as you can see, the expansion of our range of markets and applications supported by our technology. The omics and in specific to the NGS applications listed on the right are currently implemented in academic, reference, clinical, and biopharma laboratories, each of which has a distinct demands and stringent quality standard. These standards that Covaris consistently meets with precision. Speaking about enabling the whole genome sequencing for a population and performance studies, here is a case study showcasing Covaris technology in the All of Us research program, where Covaris was utilized to fragment human genomic DNA, which enabling whole genome sequencing for a large-scale population and performance studies. As many of you may know, the All of Us research program is a comprehensive longitudinal cohort study involving about 1 million participants, which is aimed at advancing biomedical research and improving health outcomes for all of us. This program has identified more than 1 billion genetic variants, which includes approximately 275 million previously unreported variants. Findings from this initiative have been extensively published in several peer-reviewed journals. A couple of them are highlighted in these sections as well. And addressing the mixed sample type workflow challenges, this has been very challenging in the current NGS space. This example highlights customer data presented by Twist Diagnostics during a Radboud webinar, where Twist transitioned from their enzymatic fragmentation to a Covaris-based, AFA-based approach. Some of the challenges with the enzymatic fragmentation, which include a wider fragment size distribution, especially when dealing with several different sample types, which is a clinical sample, as you can see here in the bottom left images. Optimizing with different sample types, which is a challenge, which was effectively addressed using a Covaris AFA technology. The robust and reproducible performance of AFA achieved consistent and tighter fragment size distribution across different sample types, which ultimately enhanced the process control, which is a crucial factor in scaling up workflows. This uniform insert size enabled cost reduction by ensuring precise coverage, with a Twist reporting about 3-fold enrichment, as you can see here shown in the right image. In summary, this data clearly illustrates how mechanical shearing with AFA significantly improves sequencing quality metrics and also cost efficiency in their whole exome sequencing workflows. Speaking about automated and streamlined library prep solutions, I would like to share highlights from a related study conducted by Tom's group utilizing a Covaris R230, which is integrated with their Hamilton automation platform to analyze several hundreds of samples. This study achieved an impressive sample pass rate over 98%, which demonstrating a highly reliable and reproducible workflow. Also speaking about sequencing metrics, which includes mean coverage, percentage duplicate reads, and reads on target, which are consistently exceed QC threshold. This streamlined and high quality workflow significantly contributes to reducing the cost per sample. Tom will provide more insights into this automated clinical rapid whole exome sequencing workflow in a while. The mechanical fragmentation of somatic workflow, which is similar to the germline solution, Covaris technology has been widely applied for somatic solutions, for example, DNA shearing from FFPE tissues for whole exome sequencing on targeted panels. In this example from Discovery Life Sciences, customer used Covaris for DNA extraction and fragmentation, observed enhanced performance compared to enzymatic fragmentation, particularly with very low input DNA from FFPE clinical samples. It is noteworthy to mention here that the discordance rate remained under 2% when using the Covaris, compared to over 15% with the enzymatic library prep at using the 10 nanogram DNA input, which is shown in this bar graph on the bottom left. Covaris in RNA-seq or the transcriptomic workflow, Covaris is well known for its AFA-based DNA shearing, often referred to the gold standard for the DNA fragmentation. In addition to DNA applications, this focused ultrasonication technology is also widely applied for RNA shearing for their RNA-seq or transcriptomic workflows. Here is one of the case studies from MIT Biology Genomics Core Lab. Covaris AFA was used for RNA shearing on clinically derived ultra-low input FFPE samples. The top right gel image highlights the highly variable in RNA sample quality. Despite this, using a single protocol, they achieved consistent shearing on inputs as low as 2 nanogram, which resulting in a highly uniform libraries, the Illumina library, regarding of initial sample quality as shown as in the gel image on the bottom right. This clearly demonstrates that AFA-based fragmentation is a highly effective across a wide range of RNA-seq protocols, producing high-quality libraries that are input agnostic. This work by MIT researchers aiming to explore the developmental origin of patient tumors, which was published in a peer-reviewed journal. Also, recently we collaborated with MIT Biology, Dr. Stuart Levin's group, to publish a technical note which further optimizes AFA-based RNA shearing for high-throughput RNA sequencing workflows. I'm excited to introduce Covaris' upcoming product, that is the TrueCover whole genome sequencing PCR-free library prep kit. This kit establishes a new standard in DNA library preparation, expanding our offering from sample preparation to sequencing. It provides a faster single vessel process, streamlining the library preparation workflow for unparalleled efficiency and precision. As mentioned here, the TrueCover library prep kit offers the simplest and fastest workflow for whole genome sequencing. Let me highlight its key features. The number one, this workflow is easier and faster workflow, that meaning a streamlined whole genome sequencing library preparation process which can reduce turnaround time up to 30%. Also, this workflow is very versatile sample input, meaning it adopts one-for-all approach for various germline NGS applications, simplifying workflows. And also cost effectiveness, which lowers per sample cost and enhances QC pass rates, making genomics research more efficient and accessible. Single tip workflow, meaning it minimizes sample transfer to deliver reliable, reproducible results. And also Covaris fragmentation, which is unbiased insert sizes, which reduces the insert size bias across various Illumina sequencers, supporting high quality sequencing outputs. This kit is designed to optimize both efficiency and accuracy in whole genome sequencing applications. Speaking about the early access program, Covaris is excited to offer an early access evaluation for the TrueCover whole genome sequencing library prep kit. We welcome collaboration and are fully prepared to provide comprehensive support. If interested, please reach out to schedule a discussion with Eugenio Deviso, the Vice President of Solutions, and also Rich Doverspike, Executive Vice President for Strategic Partnership. In summary, why choose Covaris? Covaris has long set the gold standard in DNA sharing. Our technology is recognized across the market for its superior capabilities in sample and library preparation, as well as DNA and RNA fragmentation, which is essential element for reliable NGS workflows where precise results are matter. Our solutions not only enhance laboratory efficiency by reducing hands-on time, but also ease the financial burden of sample prep. In addition, Covaris instruments are designed to meet the full spectrum of omics research needs. We are here to help solve your sample prep challenges. Please reach out to discuss your upcoming projects or to explore ways to enhance the data quality in your lab. Once again, thank you all for your time and attention. Now, I would like to invite Tom Hofste to share insights on how Covaris technology supports for their high throughput and automated NGS workflows in their core facility. Thank you.
Thank you, Madan, for your nice introduction and that I'm able to speak about our workflows. My presentation will go on with the implementation of an automated diagnostic exome and a whole genome workflow for multiple sample types. My name is Tom Hofste, and I work already for several years at the Radboud UMC as head of the core facility at the Genome Diagnostics, and I work all several years in this department. Started as technician. I have a lot of knowledge about lab automation and experience, and I want to walk through this presentation with you together, how we set up our workflows. Our department is divided in 3 different sections: the diagnostic section, the research, and the clinical section. In total, 350 employees. I'm heading the Radboud facility, the Central Genome Analysis Lab. This contains around 36 employees, technicians, and sample registration colleagues. We do around 25,000 DNA extractions a year and report 16,000 reports a year. We have a couple of different high throughput workflows, and all is automated with several devices. And all is done with a cooperation with our counterpart, Maastricht UMC in the south of the Netherlands. Now I will focus on the workflows, and I will go to the evolution of the next generation sequencing in the diagnostics at the Radboud UMC over the years. We were already in 2009, we had already some experience with next generation sequencing. We start with the Roche 454 sequencer and the solids from Applied Biosystems. And we start there also with the first experience with Covaris. In these workflows there, the Covaris S2 single sample shearing system was connected there. In 2012, we start with exomes as first tier test for individuals with intellectual disability and some other clinical and genetic heterogeneous disorders. The sample shearing was then done on a Covaris E210, a 96 format system, because the number of samples was increased already. The library performance was done with an Agilent kit, and the sequencing still on a solid system. That was our first experience with the exomes in that year. In 2014, we had a look already in genomes, and in that point, genomes were quite expensive because it's 10 years ago. But we mentioned then already, let's move to the genomes. But that took a while, and the first comparison we did in 2019 until 2021, we compared the exomes and the genomes with each other to see if it's possible to use the genome sequencing as first tier test for specific disorders. I will focus now on the exome workflow, and you can see here a diagram of 2013 until now. That are our numbers of exome sequences a year. You see an increase, and around now we do more than 10,000 exomes a year. In 2022, we start also with genome sequencing, and you see here now in this graph 2 different colors. The dark blue colors and the light blue colors. I will explain that a little bit more in this slide. Because we had a high throughput workflow from 2013 until 2019, and that workflow was library prep and sequencing, and we outsourced our samples to another center. We sent out our DNA samples and we received back raw data. In-house pipeline did the analysis in our hospital. But next to that, we had also a rapid workflow, the rapid exomes. That was a library prep and a sequencing in-house. So in the Radboud UMC, we did 16 samples a week. It was completely manual and has a turnaround time of 15 days maximum. But yeah, only 60 samples a week. We want to increase and do it all in-house in the Radboud UMC. So we developed from 2019 on a high throughput workflow for everything, for the exomes. We did automate the library prep and the sequencing in-house for 288 samples a week, fully automated with the same turnaround time of 15 days. How the workflow looks like then. We start always with the central sample receipt at the Radboud. It's Radboud Broad. The sample administration will take place, and on each sample there is one unique 1DD barcode. This one is the most important, so we can track and trace all the samples through the lab. We do, as mentioned already before, more than 25,000 samples a year, and this is also the big challenge. The main workflow is DNA extraction out of blood, EDTA blood. Next to that, we receive 21% of that is DNA from different labs over all the world. And next to that, you get 3% different start materials. And that's a challenge to get the same quality of DNA out of all these different materials or out of the DNAs that's sent in by other labs. So what we do normally after the DNA extraction, we normalize our DNAs. For the exome workflow, we normalize these DNAs to 15 nanograms per microliter, and for the genomes, we normalize to 15 nanograms per microliter. Directly during this normalization, we do also a QC SNP profiling. That's a multiplex PCR on all the samples, and we sequence them on a sequencer, and we make a profile out of 55 different SNPs. And we will store that. And we do also a quality check on gender and compare that with the sample administration if the gender is correct. And we look into this for contamination. If that's all fine, then we go forward with this sample and it will go on into the exome or in the genome workflow. When we start with the whole exome sequencing, we start first with preparation of the DNA, and our preparation of DNA is shearing of the DNA. The input is 60 to 150 nanograms of genomic DNA per sample, and that DNA can come from EDTA, cell line, fibroblast, prenatal samples, or FFPE even. And then we start with DNA shearing, and we shear on the Covaris R230. And you can see the instrument here below, and it's next to an Hamilton robot, and a small graph of a tape station profile, how the profiles looks. And we aim for 350 base pairs for the exome workflow. And after that, when we start with the preparation of the DNA, we can load 48 or 96 exome samples into the robot system, what you see here on the picture, or 32 whole genome samples we run per plate. And we start DNA shearing with the Starlet and on the Covaris R230. And in the next slide, I have a small video where you can see how the system works. plate is now in the Covaris R230, and you see the movement, and you hear also the sound of the Covaris that's making. And when this is finished, the lid of the Covaris will open automatically, and the robot is able to pick up the plate out of the Covaris and bring it back into the deck of the Hamilton starlet. Now the robot is picking up the plate. And can place it back on his carrier. Now the plate is back in the instrument and it's lidded with a lid, and the liquid handler can take off the lid and transfer the samples to the blue plate. So, and we are able to run or process 1 until 96 samples on this plate. Okay, when the shearing is done, then the next day we start with the library prep. And the library prep is from Twist Bioscience. We use the comprehensive 2.0 exome prep. We have 4 library robot stations available to run this prep with a total capacity of 384 preps a week. You can see the system on the right side. It's a Star Plus at the moment, and the robot contains a 96 head, 8 channels, also an on-deck thermal cycler for the incubation steps for the end repair ligation and the PCRs, pre and post PCR, and some heating and cooling positions for the reagents. Also some piercing tips, and we use piercing tips especially to pierce through the seal of the index barcode plate, so that you not need to peel off the seal in this process. How looks the protocol of the library prep? The fragmentation is already done by the Covaris, so this step is not performed in this station. So we start in the morning directly with the core library construction. We start with the end repair ligation, and afterwards the PCR pre-amplification for indexing. Directly afterwards, we start also with the quantification on the liquid handler, and it will pull automatically the 8 plex. The next day, the system will hybridize, or overnight the hybridize will take place, and the next day the pre-washing and the streptafidino wash will take place, and the post-purification and post-amplification will take place on the same instrument. When that is finished, then all the samples will be pooled in one tube, and we will sequence them on the Illumina NovaSeq. And at the moment, we sequence the exomes still on the NovaSeq 6000. And how will this look onto in the instrument? Here is an example of one flow cell. We run 48 WES samples on an S1 flow cell and 96 exome samples on an S2 flow cell of the NovaSeq 6000. In 2024, we had a first-time write of 96.4%. When the prep fails in the first time, we do automatically a repeat of the prep, and the second time write is 99.4% at the moment. So only 0.6% is failed in this process at the moment. If we focus then a little bit more on the results, on the sequencing metrics, you see here in the upper graph the coverage, the mean coverage of all the samples. The dark blues that are fine. We aim for 80x coverage for the samples. The light blue samples are below, and they need to repeat it for prep and sequencing. Below you see the on-target percentage. We are around the 65% of on-target. And the above slide is the insert size. You see here the inserts around the 500 base pairs, and that's at the moment fine and it's quite stable. You see a slightly increase in duplicate reads after a certain period, and that we can explain that because the loading concentration on the flow cells is changed, why we have now a little bit more of duplicate reads. You have seen the QC SNP profiling after the normalization. We did the 55 SNPs after normalization, but we will take also this 55 SNPs out of our exome data and we make a comparison. If it's matching, then we know exactly, okay, this is still the patient and we can start reporting. If we see it's mismatch, then we know there is somewhere a sample swap. So then we stop directly and we need to complete everything again. So then we are secured with this test that patient is still the patient. Okay, this was the exomes. Now I want to move to the whole genomes because we started in 2022 with the genomes, but in 2023 we start with the automated library prep. In this year we run a little bit more than 2,000 genomes, and in 2024 we are around the 5,000 genomes at the moment. You see an increase in this graph, and then big red line, and the big red line is the implementation of the NovaSeq X Plus for 1 month ago exactly. There was only one rule for the whole genome workflow, and that was the library prep needs to be PCR free. In the first beginning, we start with DNA shearing with segmentation, and later on we step over to DNA shearing with the Covaris for mechanical shearing. At the moment, we have 2 library robots available for this prep with a capacity of 192 preps a week. This robot has only 8 channels, an on-deck thermal cycler for the incubation steps. It's PCR free, but there are still some incubation steps. Heating and cooling positions for reagents, and also piercing tips to pierce through the index adapter plates. As mentioned, we prepare first the DNA. For the segmentation, it was directly in the library prep robot, but now when we change to the Covaris, we do exactly the same as for the exome workflow. We prep the DNA before. The input is 350 until 500 nanograms genomic DNA per sample. At the moment, only for DNA from out EDTA blood. And you see here below a graph of the tape station with a mean size of 400 base pairs where we aim for. And why we do use only DNA out of EDTA at the moment? Because we had done a study, and this study is done at the moment only with the segmentation kit. You see here all graphs from different materials. The most upper part is our main workflow, the EDTA workflow, where we had more than 1,500 samples in this study, and the other ones are all different sample materials. You see here the average autosomal coverage, and you see that different materials will drop below the 25x, the red line, and the 25x coverage is where we aim for. When it's below, we cannot report. So there is for us a too high risk that it doesn't reach the 25x and that we need to repeat it. So in this time we decide, okay, this workflow can only be for EDTA at the moment based on the segmentation kit. We look also into the insert length for this set of data. Yeah, and you can see that there is some difference, but not that big difference. So we expect no issues for the insert size at the moment for the segmentation, but we need to repeat this complete test also with the Covaris and the library prep that we use at the moment. When you look to the uniformity of the coverage for the whole genome, it's fine for all different materials. So we expect also no issues there. why we changed then from library kit and why we changed to the Covaris shearing. With the current or the formal kit, we had some low library yields. We had less influence on the insert size because it was segmentation and you cannot turn that many based on the insert size. And we want to use multiple sample types, and we believe that with the new kit we can use multiple sample types and we have more influence on the insert size. How will it look then when we focus on the results? You see here 2 copies of runs. In the top of the window you see the 24 whole genome samples on an S4 flow cell of the NovaSeq 6000 with the Illumina DNA PCR-free kit. We did in total in 2024 more than 5,000 samples, 2.9 fails in library prep and 4.5 fails after sequencing. And fails after sequencing is less coverage than 25x. And these are only samples of DNA out of EDTA. And below our new workflow, what is only live since 1 month, it's 64 whole genome samples on a 25B flow cell with the NEB Ultra 2 DNA PCR-free kit. And in 2024, we did 512 samples, 0% fails in library prep and 4.1 fails after sequencing, also drops below the 25x. So when we focus then more a little bit on the quality metrics of sequencing, when we look to the coverage of the genome, the lower part below light blue is below the 25x coverage, sorry, and we see, it's quite stable. And if you look to the duplicate reads, you see after the vertical line, so the implementation of the NovaSeq X, a slight increase in duplicate reads, and that's more or less sequencer dependent. When we look to the insert length for the segmentation, it's quite stable on EDTA workflow. When we took our NovaSeq X in production, you see a decrease in insert length, but that's NovaSeq X related. And we are quite happy with the Q30 base quality value. We see definitely an increase of 4% on quality Q30 basis. What we'll do in the next future, we don't know exactly. We know the numbers that we have run in 2023 and 24. You see a slight decrease in 2024 for the exomes, and you see an increase in the short read genomes. And we know already that we run in 2024 also 250 optical genome mapping samples. And we will increase that in 2025 to 500 samples a year. But also we will start with our first long read whole genomes in diagnostic. But what will happen with the short reads, we don't know exactly. But we can imagine that the short reads exomes will decrease and the short reads genomes will increase, and then in competition with the long read whole genomes. So a question mark for us as well. Now, how it will look then in our laboratory for hardware, we have a couple of liquid handlers from Hamilton, 2 Covaris systems to have a backup in the diagnostic field, a couple of Illumina sequencers and PacBio sequencers for the long reads, and also some BioNano systems for the optical genome mapping. So this was my presentation, and I will thank different teams, and especially the Radboud UMC team, to make this all possible in my lab, and of course all the companies that are working with us to make it happen. So thanks, and when you have questions, please welcome.
All right. Thank you very much, Madan and Tom, for those great presentations. We'll now start the live Q&A portion of the webinar. So if you do have any questions, make sure you go ahead and get those in now. We're going to start working our way through the ones we have, and we'll answer as many as we have time for. So for the first question, Madan, we'll start with you. What are the fragment sizes and sample volumes supported by the Covaris shearing?
Yeah, I can take this. Thank you, Matt. Covaris shearing is optimized for a wide range of fragment sizes from 150 base pair to 3 kb and even up to 5 kb. Regarding the sample volumes, our AFA-based consumables are designed to accommodate volumes ranging from 5 microliter to 130 microliter, making them suitable for most of the NGS library prep workflows available in the market. If there are any specific questions related to your custom workflows, please reach us out. Covaris Solutions team will be happy to assist. Thank you.
All right, the next question we have is, can you comment on how difficult it is to integrate the R230 Covaris instrument into the Hamilton liquid handler?
Yeah, I can comment on that. Yeah, of course, you need some experts from Hamilton to implement it next to your instrument. So you need to have an extra driver to address the correct commands to the Covaris. What we did, we test manually first all the Covaris settings on the R230 and then transfer it next to an Hamilton Starlet. And in combination with Hamilton, we make some protocols to run it together on the Starlet and on the R230. So it's quite straightforward.
Yeah. What types of samples are compatible with the Covaris TrueCover library prep compatible with?
Yeah, this is a very good question. The Covaris TrueCover library prep workflow is currently compatible with both germline and somatic samples, including DNA isolated from blood, saliva, and FFPE tissue samples. Thank you.
All right. Let's see. In your automated WES workflow, you achieved a first-time success rate of 96.4%, followed by a second preparation step for the initial failures, bringing the overall success rate to more than 98%. Could you elaborate on the adjustments or strategies implemented during the second preparation to minimize the failure rate?
Yeah, that's a good question. What we do if in the end the library is not succeeded, we do a second time repeat automatically on the prep. And the only thing that we do, we start with a new aliquot of DNA, make a new normalization sample, and we start again the shearing on the Covaris, and we do a complete re-prep of the sample. Because the complete workflow is that quite long for the exomes, that it could happen that it goes somewhere wrong. So we repeat completely the sample and we see that success rate at the end.
Matt, I think you are on a mute.
I apologize for that. Could you comment on how the Covaris shearing and library preparation workflows facilitate the standardization of insert sizes across different sequencing platforms?
Yeah, this is an excellent question. Covaris achieves precise and consistent fragmentation, which is complemented by optimized size selection and also proprietary magnetic bead based reagents. Together, this ensures uniform insert sizes regardless of the specific Illumina sequencing platform, whether it is a MySeq, NextSeq, or NovaSeq X or X Plus. Please feel free to connect us if you would like to learn more about TrueCover library prep workflow.
Got a little multi-part one here. Are you able to use the Covaris 96 AFA tube TPX plate directly on the star NGS, on deck thermal cycle after fragmentation, or do you transfer the samples to some other plate? If so, what plate? And how much sample loss did you observe, if any?
Yeah, I can argue. I can answer this question. What we do, we don't use the Covaris plates in our PCR devices or on the star NGS, because we run not always complete full plates of the Covaris plate for shearing. So then you have positions lost in your Covaris plate. So we choose in this way to run the shearing in the Covaris plate and bring the samples over to a new plate, and that's a full skirted plate. And I'm not sure which plate. It's a standard PCR plates that will fit in the on-deck thermal cyclers. There is a little bit of sample lost, but we take always, I think, 5% of extra DNA into the Covaris plate and transfer as much as possible to the PCR plate. I'm not sure if Madan can argue if the plate is okay for on-deck thermal cycling.
Yes, Tom. Yeah, I can supplement that. Yes, the AFA TPX plate is compatible for on deck, and we are closely working in develop even for the plate sealing as well.
All right. Got time for one or two more. So if you do have another question, feel free to send those in. The one here is for your full genome sequencing workflow. You initially used Illumina tagmentation before transitioning to Covaris. Could you elaborate on the challenges you encountered with the enzymatic fragmentation?
Yeah. Of course, it's a tagmentation on an automated platform. So there's always a challenge to get the correct amount of enzymes into the wells and a correct mixing. So it's the same challenging steps as a normal enzyme reaction. So and of course, the time for incubation is critical in this way, and the way of addition in which order you add the components. So that was the challenging part of tagmentation during our work flow for the whole genomes.
All right, excellent, thank you. So I think that's all we've got time for today. Thank you, Madan and Tom. Do you have any final comments you'd like to say for the audience before we wrap things up?
Yeah, I can go first, Tom, if it is okay with you. Thank you all for taking the time to listen to the webinar. I hope this presentation provided valuable insights that you can take back to your work. Feel free to reach out if you have any further question, feedback, or we are happy to take ideas to discuss. We'd be delighted to continue the conversation. Once again, thank you for your time, and have a great day.
Thank you. Thank you again to Madan and Tom for their time today and their important research. We'd like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Before we go, I do like to thank our audience for joining us today and your questions. Any questions that we didn't have time for today, and any of those submitted when we have it on demand, they'll be addressed by the speakers via the contact information you provided at the time of your registration. If you'd like to request additional information, please be sure to fill out that Q&A there, and someone will be reaching out to you directly. The webcast can be viewed on demand, and LabRoots will alert you via email when it's ready for that replay. And we do encourage you to share that email with your colleagues who may have not been able to make today's event. And until next time, thank you very much, and goodbye.
Thank you all.